We'll turn it over to Steve's presentation. Thank you.
Hello, my name is Steve Hunter, and I come from a small village called Grant Park in the state of Illinois in the farmlands. And after a very long journey, I've ended up in the city of Portland, Maine, a city of harbor and seagulls and oysters. During that long journey, I did spend 5 years in the University of Wisconsin in Madison, studying under Tom Givnish, where he introduced me to the Hawaiian Lobeliads, and as my thesis, I created a molecular phylogeny of the Hawaiian Lobeliads. As I said in the introduction, there was a problem with that in that we had outsourced the assembly of the nuclear genes, and they had not taken the tetraploidy into account correctly. So the last 2 years, that's what I've been doing, is reassembling the nuclear genes and then reanalyzing all of the data. And so today I hope to show you that phylogeny, and also I will be discussing the methods that I use to generate the phylogeny and pointing out some of the interesting patterns and other hybridization events that occurred. So without further ado, let me start introducing the phylogenies that we found for the Hawaiian Lobeliads. So let's start out with the backbone of the Hawaiian Lobeliads. I have 6 points to make about this. First, all 8 of the genera and sections of the Hawaiian Lobeliads are reciprocally monophyletic, meaning there's no current hybridization between them. 2, we have very strong support that Galeotella and Campanulaceae are sisters, Ricamia plus Delicia are sisters, and that the 2 Cyanea sections and Claremontia are sisters. 3, we actually don't have very strong support as to the relationships between Galeotella plus Campanulaceae, Ricamia plus Delicia plus Revolutella. It's even quite weak whether those 3 together are sister to Cyanea. We have very little support as to the position of Sclerotheca, whether it is inside the Hawaiian Lobeliads or outside. Also, we now have very strong support that it is the purple-fruited Cyanea that is sister to Claremontia, and together those are sisters to the orange-fruited Cyanea. Okay, on the Campanulaceae. So for the rest of the presentation, I will be using this gray, meaning we didn't include the genetics in the study, and so those are based on geography or prior studies. So for Campanulaceae, you see we've got nice progression row with Hawaiiensis down here as the first branch, and then Singularis and Kalai, both from Oahu, and then Macrostachys from Maui Nui. So I placed these 2 extinct Campanulaceae from Maui Nui together with Macrostachys, based only on their geography, and then here are your 2 Big Island species, which I placed as sister to the Maui Nui species. Okay, Galeotella. Very similarly, we've got the progression rule with our Kauai species, sister to the Oahu species and the Maui Nui species. There are no Big Island species. The most interesting thing here is that Lammers created this Lobelia cross Hawaiiensis, which he considered to be a hybrid between Velosa and Wakiawa. We did not. Our sample of Hawaiiensis is actually quite old before Lammers renamed everything, so whether that is Wakiawa or Hawaiiensis, we don't know. But in any case, with only 2 species in the sample, we could not say anything about whether Lammers was correct on this hybridization issue. That will require further study. Delicia, a rather sad genus because 11 of the 15 species are extinct, but we did managed to collect the other 4 extant species, and the other progression rule exists. So the 2 Kauai-stensis, Rotidosperma and Kauaiensis, are sister to the remaining taxa, which we had Delicia hawaiiensis from Oahu and Delicia arrutidentata from the Big Island. The most interesting thing here is when Lammers revised this genus, he created 3 sections, and one of those sections he actually put arrutidentata with Kauaiensis, which our new data is showing that is not possible. So those sections are not valid. Yeah, a small genus with only 2 species, and therefore we really can't say very much about them. We did find them to be sister. There was no hybridization with any other. As we mentioned earlier, Ricamia is sister to Delicia, And the age of this split here is old enough that they do appear to be good species, both genetically and morphologically.
Those first four clades were quite simple. There were no hybridization nodes, and they did not contradict the progression rule. As we move into Revolutella here on the left, you'll see we are starting to get hybridization nodes, and things are getting a little more complex. Let me get off the screen so I can describe what's being shown here. Here we have two hybrid nodes. The first of them is Lobelia eucoides from Oahu, which I hypothesized to be a hybrid between the Kauai accession of eucoides and this other clade, which includes Lobelia nihoensis. There are, at each hybrid node that I'll be showing, there are two symbols. The first of them is this, which is the symbol for Venus, I believe, which is where the plastome comes from. So this is the maternal inheritance. The other two are these gamma figures, which tell which percentage of the nuclear genome comes from which of the parents. So in this case, I hypothesized that the plastome came from Lobelia nihoensis, and that the nuclear basically came from Lobelia eucoides. This is often called chloroplast capture. The other hybrid node, this one, is Lobelia hillebrandii, which now I believe the plastome is coming from the Oahu eucoides, and the nuclear is coming from Grayani dunbarii clade. In order to create that hybrid network that I showed on the previous slide, I used a technique called quartet analysis. Quartet analysis, basically the quartet comes because you choose 4 species at random and then apply Bayesian analysis to that. How you choose those 4 accessions depends on what you're trying to show. For example, in this case, let's say I'm trying to show that these 4 Lobelia hypoleuca species form a monophyletic clade. And so to form the quartet, to do this particular exercise, you choose 2 of these accessions, for example, 19605, 26271. Then for your third species, you choose something from these 9 Revolutellas outside of that clade. And for your fourth species, you choose something outside of Revolutella. Now, I have 100 nuclear genes for my data. And so I'm going to run the 4 accessions that were chosen, a Bayesian analysis on each of those 100 genes. Now, when you run Bayesian analysis on a gene, on a quartet, there's only 3 possible trees. So one possibility is that the C plus D accessions are closest sister. Another is B plus D are closest sister. And the third possible tree is that B plus C are closest sister. Bayesian analysis will give you a probability of these 3 trees. For example, if Bayesian analysis really knows nothing and all 3 are equally probable, it'll give you 33% probability. So you get a point there. If instead this tree, C plus D and B plus D, were equally probability, 50-50, and this was not probable at all, you'd get a point here. If this tree was 50% probable and these 2 were equally probable, you get a point right around here. But for each quartet, you'd get a single point with what's called a concordance factor, which is what percent of the genome points to this tree, and this tree. Now, what you can see here is when I've run this to see if this clade is monophyletic, every single point showed up here in this apex where C plus D, the 2 hypoleuca trees, the 2 hypoleuca accessions are closest sister to each other. So each point here is a quartet. So that's right. So I choose all possible quartets. Now, this is a huge amount of work. So I've got 2,000 quartets, 100 genes. So that's 200,000 Bayesian analyses. And so luckily now there's this center, Wisconsin has a center of high throughput computing, which has access to 20,000 cores in it. So it uses massive parallel processing. Now here's another example. Now when you see these gray points over here, those are points in which Hypoleuca is being compared to any of these other species. And you can see there's quite a bit, it's aiming towards the center here, which means lack of resolution, but it is very symmetric around the axis. And I interpret this as that means Revolutella was a very quick break from Eucoides, but there was no hybridization. If there was hybridization, you would see an asymmetry in these points. They would be pulled towards preferring this tree or this tree. But since there is none, that means it's quite random. Some genes say one way, other genes say the other, and we interpret this as incomplete lineage sorting. So what might hybridization look like in a quartet analysis? Well, here this has nothing to do with Revolutella, but just to show an example. Both of our accessions of Cyanea lobata appear to be hybrids between the Platyphila and the Grimesiana clade. So what do we have here? Well, in our bin D is where we've put our Cyanea lobata. And these red points represent Platyphila when compared to Grimesiana. And so in that case, the B clade is represented by down here at this apex, and the C, which is Platyphila, and the C clade up here, which is Grimesiana. Our Cyanea lobata appears to be halfway between them. Our Cyanea lobata appears to be half Platyphila, half Grimesiana. These blue-green points are actually glabra. And so because Platyphila and glabra do appear to be sisters, they share a lot of genes, and so our Cyanea lobata shares genes via Platyphila from glabra. Therefore, the signal's not as strong as glabra, but it certainly is there. So when you see this asymmetric pull, it's something that's not on this main axis. When you see it's pulled away from the axis, that's usually a sign of hybridization. Finally, here's an example of what it might look like if we're doing a quartet analysis and the results are inconclusive. So here, back to Revolutella, I've got Dunbarii, Grayana, and Hillebrandii being compared to each other. And as you can see, all the points are centered here in the center of the graph. There's a small pull towards B and D being sisters, B being Dunbarii and D being Hillebrandii, but they're so close to the center that I would call these results inconclusive. So how do we go from the quartet analyses to the hybrid network? Unfortunately, that is still, in the work that I did for the Lobeliads, was still a bit of artwork. I didn't use a program that came up with a maximum likelihood type of calculation. Rather, I used the data and some deductive techniques to determine where I needed to place hybrid nodes. I always started with the plastome. So up here is the plastome tree. Now, the plastome is it's not the best, it is just one of the many possible evolutionary pathways that the Revolutella took. And so I usually start with that, and you'll see that if I come over here and cross out these lines where the plastome did not go, you'll see that this then turns back into this plastome tree. Your first branch is Lobelia eucoides with sister to Hillebrandii. Your second branch is Eucoides of Quai, and then your next branches are Hypoleuca oahuensis, Grayana plus Dunbarii. But that does, so that's the tree that is the plastome, but that does not line up with what we're seeing in terms of the quartet analyses. So we saw that we did see this tree at one point. So eucoides together seem to form a clade, and everything outside of eucoides forms a clade. And so in order to make that happen, I claim that the Eucoides on Oahu is a hybrid between the Eucoides on Kauai, and it got 100% of the nuclear genes, but none of the plastome. And so that's how those two ended up being sister in the nuclear genes. And then Hylobrandiae actually does line up with the Dunbarii and Grayana. How did that happen? Well, while it still has the plastome from the Oahu Eucoides, well, that's because it is a hybrid between Grayana Dunbarii and the Oahu Eucoides, where it picked up the plastome and 100% of the genes from these two. And so it was that technique that I used to turn this quartet analysis and the plastome phylogenies into these hybrid networks. Now, there is an alternative approach to the quartet analysis, which I use to discover the hybrid network. One of them is the species tree approach. Now, in this, a species tree is when you take the nuclear genes and you apply some methodology to generate from them a single tree, a bifurcating tree that possibly represents the dominant characteristic. So I've used here the population and primary concordance tree, which is a species tree, and here it is shown here in the bottom left. The population tree and the primary concordance differed in how they ordered Hylobrandiae, Dunbarii, and Grayana, so I just showed them as a polytomy, but each of them did show it as a bifurcating tree. And you'll see that this species tree puts the Eucoides together, and it puts Hylobrandiae with the Grayana and Dunbarii. And here's the plastome tree, which we'd already seen, which put Hylobrandiae with the Oahu Eucoides. And looking at this, you could, similar to what we did with the quartet analysis, say, what do we need to do with these two trees to create a coherent hybrid network? And you'll say, well, Hylobrandiae needs to go over to this group in order to match the species tree. And we need to do something to get these two Eucoides together so that they match the nuclear tree. And you could, from those two pieces of information, generate the hybrid network shown on the right. I do wonder, however, if this species tree method can be used for a more complex network. Here I'm showing the network that I discovered for Claremontia using the quartet analysis. And as you can see, I found up to 7 hybrid nodes. So let's look to see. So here's the plastome tree on the left and the species tree on the right. And as you can see, the difference, one important difference is that Claremontia arborescens has moved down to the tuberculata peliana clade. So in the plastome, it was found in this clade, and in the species tree, it's found down here. And then similarly, the ordering inside the Pallidamicranthon samuelii has changed. So from these two differences in these two trees, you might be able to place two hybrid nodes on a hybrid phylogeny. And sure enough, actually, Micrantha was found here using the quartet analysis method, and arborescens actually was involved in quite a few hybridization events. But there are other nodes here that weren't discovered, didn't show up in the species tree method. Here in the oblongifolia and persicifolia, this node wasn't found, and possibly the reason is that a dominant part of persicifolia, both the plastome and possibly 70% of the nuclear gene, comes from oblongifolia, so it appears quite close, and you don't catch that difference. But when we look at the quartet analysis, we see this unusual asymmetry when we do the study, which points to a hybridization event. And similarly, Claremontia peliana had the same position between those two, between the species tree and the plastome tree. But when we did the quartet analysis, we found it had this unusual relationship with the Big Island Claremontia, which pointed to a hybridization node between those two clades. Another issue with species tree methods is that they appear to be inconsistent. In 2016, Claudia Solis-Lemos in Cecile Anne's lab in Wisconsin ran a study in which Claudia simulated a hybrid network, and she put in one hybrid node, sometimes giving it a gamma of 10% and sometimes giving it a gamma of 30%. And then she simulated from 10 to 10,000 nuclear genes and ran Astral on the simulated sequences. And when she was using 10%, all was good. White represents the dominant tree from the hybrid network, the dominant being the tree formed by the 90% of the nuclear genes. But when she ran it with a gamma of 30%, she got quite a different result. No matter how many genes she added, the percentage of time she discovered this dominant tree never increased. And importantly, so you have the dominant tree where here 70% of the genes come from, but you also have the alternative tree where 30% of the trees come from, and that is represented by this black line, which means all of these tan and green lines represent trees that do not represent the true evolutionary history of the simulated sequences. Another benefit of the quartet analysis is the ability to scan across many hundreds of different accessions looking for signs of hybridization. In 2013, Givnish et al. Published this paper on Clermontia, and among others, he proposed that Clermontia tuberculata, rationally so, is a hybrid between arborescens and aculeata flora. Clermontia tuberculata is the only Clermontia with prickles on it. It is found sympatric with aculeata flora, and it made sense that it could be a hybrid. We were able to search all possible combinations of tuberculata with all the other cyanias, and we found no signs of hybridization. Again, the work that Tom did on this was based on very little data that was available at the time. We have much more data now. This then is the reason that the hybrid results I'm showing you today have not yet been published. I have not yet been convinced that the species tree method is the correct to use on hybrid networks. At best, I can say is that the species tree methods are inefficient. So they pointed towards 2 hybrid nodes in the Clermontia, whereas I believe there are many more. And at worst, they're actually misleading. As Claudia's study showed, more than 50% of the trees that came out of the species tree method were incorrect, not true evolutionary pathways. Unfortunately, it is common today to use the species tree method, and the senior advisors on this project are insisting upon its use, and I'm being stubborn and don't want to use it, and that's the reason we haven't published yet. But I beg anybody out there to convince me that using this method is correct, and I will, once convinced, I'll be happy to use it and then finally publish these results. On to the purple fruited cyania clade. I have 3 points I'd like to make here. First of all, here is Clermontia pyrularia. Here I've labeled it as Cyania pyrularia. All evidence points to this being pyrularia being a Cyania and not a Clermontia. 2, I am proposing a hybrid network with a rapid speciation at the base and then 2 hybrid speciation events, one of which caused the Superba clade and the other which caused the Coriaceae clade. Finally, Pyrularia and Leptostegia have been described with yellow or orange fruit, and everything else has purple. So here is probably where the purple fruit originated, and then it made its way via this hybrid node to get onto the Superba clade, and on this hybrid node to get onto the Coriaceae clade. And the final point is that in the Coriaceae clade, there does seem to be some east-west structure. Here's Clermontia, which we've already seen briefly. I've got 3 points I'd like to make on this slide. 1, you will not see Clermontia pyrularia. All evidence points to that species as being part of the purple-fruited cyania. You also won't see here Clermontia grandiflora, and that's because we had 5 different accessions, each collected by an experienced collector, a different experienced collector, and each of those 5 paired with a different species. So one of them paired with Pelliana, another paired with Pallida, another paired with Tuberculata, another with Arborescens, and finally another one with Kakiana. So I don't know what's going on with grandiflora. I think this would be an excellent place for a study to make sure that if there's some kind of identification problem, or is there rampant hybridization. And finally, the orange-fruited cyanea. I've broken this down into a series of slides because of the number of taxa within this group. We found 6 large clades, which I've just labeled Rolandia, Grimesiana, Platyphila, Glabra, Pilosa, and Hertella. We do see that Glabra and Platyphila are sister to each other. And we had 2 accessions of Hawaiian lobata. Both of them appear to be hybrids between the Grimesiana clade and the Platyphila clade. Whether that's true for all lobata requires further study. Now here I've broken up Rolandia and Grimesiana. So here's Rolandia, the core Rolandia, and here's the core Grimesiana. Within these, I found very little structure, so I'm not going to try and determine explain relationships within them. But very importantly, between these two, Pinatophyta truncata and possibly St. Johnii and Konahua nuiensis are all hybrids between these 2 clades. Here is the Platyphila clade, and here is the Glabra clade. Don't have too much to say here. The tall Hawaiian species, we saw very little genetic structure between them. That'll probably require population level study. In Glabra, we did see a split into 2 clades. One includes Meckledownii, Glabra, and Meriti, and the other includes all of the others. And finally, here is the Pilosa clade and the Hertella clade. This Cyanea copelandii, its position, its placement is very much in doubt. It might even go further up the tree, but the majority of evidence pointed to it to be part of this Pilosa clade, but that's still quite uncertain. The Hertella clade also had many of the species. We didn't see very much genetic differentiation between them. And so that finishes up the tour of the phylogenies that come from this study. So the phylogenies that I've been showing during this presentation have been posted into the workshop folder, and anyone that wants to download it should be able to. And I really do welcome any inquiries as to that hypothesis. My email is in that document. Those inquiries can be, challenges to the results, or questions about my methods, or about the accessions that we have, anything. And I also really was serious. I do welcome someone to try and convince me that using species tree methods on hybrid networks is appropriate. And if so, that'll be removing a major stumbling block to getting these results published. Before leaving, of course, I'd like to thank everyone who's helped me during this project. Over here on the left, I've listed the names of everyone who's helped me collect samples, who helped me with the lab work, who helped me with the computer work. Down there on the bottom right are the people who helped fund this entire presentation. Of course, I'd like to thank you all for listening, and I'd like to thank Lauren for inviting me. I've really enjoyed the conference so far, the workshop so far, and I'm looking forward to the rest of it. And otherwise, again, thank you very much.